Hello everyone, good morning. My name is Yumei. I'm from Shanxi University, China. My collaborator is Professor Shi Zhou from South Cross University, Australia. We are very pleased to be here to join this forum. The topic of our presentation is metabolomic responses to endurance exercise intervention in rats and chronic unpredictable mild stress. Our presentation will include these four parts. First, some background information about this research. Second, the main work we have done. Third, some conclusion we have got, and the last, innovation and future studies. Why we choose this item? Depression is a common illness, with more than 322 million people affected by it globally. The World Health Organization announced that depression is the leading cause of disability worldwide and a major contributor to the overall global burden of disease. The current treatment for depression is mainly by medication. Although the medication can ameliorate depressive symptoms, many patients are unable to continue with treatment because of side effects. Medication can cause some adverse reactions and has a low recovery rate after stopping medicine and a high relapse rate and a high cost. It is known that exercise intervention can improve the depression, improve physical health and fitness, and with few side effects and low cost. So, exercise therapy of depression is an important area of research with broad application potential. It is known that exercise interventions can improve depression. However, what is the mechanism of the effect of exercise on improving depression? Exercise has shown antidepressant effects. However, their underlying mechanisms have not been fully elucidated. On the one hand, previous literature has focused more on the effects of exercise on depression at the molecular biological level, such as monoamine neurotransmitters, neurotropic factors, and immune factors. However, less attention is given to the potential peripheral mechanisms. For example, the metabolites changes in urine, skeletal muscle, and serum. On the other hand, there is a veritable type of exercise that can be used in depression therapy. Why and how these interventions can result in similar or different antidepressant effects are interesting questions from the viewpoint of metabolism. Metabolomics is an emerging discipline that studies the overall changes of metabolites in cells and the organism. The picture shows the area of metabolomics application, including disease diagnosis and research, drug development, biology, etc. Metabolomics is a relatively new omics method and provides a wealth of information about metabolic changes in exercise. With metabolomics in sports science, we can explore the pattern of change in the endogenous metabolites spectrum of an organism under the influence of exercise. Various analytical techniques such as nuclear magnetic resonance, NMR, GC mass, LC mass, has been widely used in metabolic research. Compared with GC mass and LC mass method, NMR is preferred by many researchers because of its inherent advantages, such as easy of quantification, no destructiveness to samples, and unbiased metabolites. All in all, the scientific questions of the study as follow. Firstly, which exercise type has the best antidepressant effect on depressed people? Secondly, what metabolites and metabolic pathways are affected before and after intervention with different exercise types? Thirdly, how does exercise regulate metabolites in different tissues of the body? Are there similarities and differences? The meaning and aim of this research contains three parts. First, to identify potential biomarkers in metabolic pathways that were associated with the development of depression. Second, to explore and compare the potential mechanism underlying the effects of the endurance and resistance exercise in improving depression symptoms through the pathway analysis. Third, to examine the metabolomic changes in urine, skeletal muscle, and serum in response to the exercise, using the HMR and LC mass metabolomics technology, respectively. Just like other research, animal models have been explored as an important tool for studying the pathogenesis of depression. Among them, the rat model with chronic, unpredictable mild stress can be mimics many of the core symptoms of human depression and is the most effective and commonly used depression model. The validity of modeling was evaluated by the changes in body weight, sex preference test, and the number of crossing and standing in an open field text. Okay, let's see the pictures briefly. 32 male SD rats were randomly divided into 4 groups, with 8 in each group. Blank control group, CUMS control group, CUMS with endurance exercise group, and CUMS with resistance exercise group. For exercise protocols, the picture above shows the endurance exercise program, and the picture below shows the resistance exercise process. They are all classic animal exercise programs and are widely accepted. These two sliders show the cost of preparing the samples for NMR and LC mass experiments. According to different procedures, urine and skeletal muscle were used for NMR experiments, and the serum was used for LC mass experiment. The analysis process of Metabolomics contains three parts. NMR spectrum were used to identify the main chemical composition. Multivariate statistical analysis was used to look for potential biomarkers, and the pathway analysis explained the metabolic pathway changes caused by the modeling. Analyses were performed using SPSS statistical package. Two-way ANOVA and the t-test were used. PCA plus DA or plus DA were performed using CMAC-P multivariate statistical analysis software. After the 4 weeks of modeling, compared with Group C, the body weight, sucrose preference rate, and the number of crossing and standing were significantly lower, indicating that the CUMS depression-like model was established successfully. The pictures showed that both the endurance and the resistance exercise could improve the behavior indices of depression in rats on the CUMS, with the endurance experience appeared to have a better effect. The main work we have done can be summarized into 3 parts, as shown in Table 1, namely, the studies of urine metabolomics, skeletal metabolomics, and the serum metabolics of exercise. Antidepressant based on NMR technology and LC-MS technology in a rodent model of depression. 31, 32, 26, and 21 endogenous metabolites were identified in urine, gastrocnemius muscle, soleus muscle, and the serum of CUMS rats, respectively. After further analysis, 21, 10, 13, and 21 potential biomarkers were screened out, which involved 8 different metabolic pathways. The endurance and resistance exercise as prescribed in this study could recall the changes in different metabolites. Overall, the endurance exercise appeared to have more extensive effects on metabolites in skeletal muscle. As shown in Figure 3, correlation analysis showed that the body weight, sucrose preference rate, and the number of crossing and standing were strongly correlated with levels of differential metabolites in serum. The following are the main conclusions of our research work. 1. Endurance and resistance exercise resulted in common effects on the metabolic pathways of alanine aspartate glutamate, TCA cycle, and butyric acid. Endurance exercise also had effect on glycolysis or gluconeogenesis and pyruvate metabolism, while resistance exercise had additional effect on intestinal microbiome metabolism. Two, a number of metabolic pathway in skeletal muscle were involved in the improved responses to stress, and the endurance exercise might have a broader effect. 3. Combined with the result of NMR experiments in our previous report, the effects of endurance exercise on depressive were mainly related to the changes in pathway of energy metabolism and amino acid metabolism. We also find that the changes in serum levels of metabolites in CUMRS rats were closely related to behavior disorders. Innovation and future studies of our work. The current study provides not only potential diagnostic biomarkers for depression and the antidepressant effects of endurance exercise, but also a metabolomic approach to investigate the underlying mechanism of exercise intervention in the management of depression. In the future, the combination of targeted metabolomic and new technologies will be a new direction in the study of exercise antidepressant mechanism. Funding. This work was supported by the National Nature Science Foundation of China and the key research and development projects of Shanxi Province, China. Acknowledgement. We would acknowledge the research team and the co-authors who contributed to this study. 乙甲、熊薛、香玉流、俊生田、雪梅琴 and An Ping Chen. Thank you very much. Thank you.